Thank you, Bala, for the kind introduction and also for the kind invitation for this particular lecture series. What I would like to do today is give you a glimpse of the kind of research that happens in my lab, but I'm also going to give you a brief perspective about epigenetic regulation and the state of the research as of today globally. And towards the end of my lecture, I'm going to give you some highlights of most recent discoveries that will somehow, I hope, will change your understanding and perspective on health and disease in general. So the picture that I've used for the first slide is something that is the centerfold, I would say, for this talk, and I'm going to talk about what this structure is. For some of you who have heard a little bit about DNA, would immediately recognize that what you see here are the spiral, the two helices of the DNA. But what's this? Okay, this is something new because this structure, this kind of assembly of DNA inside each cell of our body is actually what is important for today's discussion. So the first picture that I'm going to show you is something which is again very unique, and it's very close to me because the first time when I saw this, I was quite, I would say, stunned completely. Maybe most of you won't be able to see the numbers, not able to read them, but this stack of books that you see, which is probably as tall as this auditorium here, and the number of volumes here actually represent something very unique. So if I were to read the numbers, this is number 1, 2, 3, 4. Of course, there are multiple volumes for each of these numbers, and it goes up to 22, and then there is X and Y. So some of you at least can, based on this, immediately some of you are nodding, saying that maybe yes, this is something to do with the human chromosomes. And yes, it is. But then why are they numbered like this? So what you see here in this stack of books actually is a single print of our genome. Okay? And the reason why this picture is important is because it tells you about the volume of information that we carry in every cell of our body. Okay? And the way it is printed here, this is actually in the Public Museum, okay, in London by the Wellcome Trust, which funded some of my research earlier, and they funded almost half of the Human Genome Project, and therefore they have kept it as a public exhibit. It's worth seeing. I mean, when I saw this for the first time, despite being a researcher in genetics and epigenetics, I was taken aback because this is at my eye level. I open this book and I start reading it. This is the information I gather. Okay? This is just from one page. And you can see that they have not left much space on the sides. Okay? There's only hardly 1 centimeter space on each side. The font size actually is very low. It's about 9 font. So it's not that they have used a big font and a lot of space and so many books have been printed just to occupy so much space. No, they have tried to actually maximize the amount of pages and number of articles, alphabets that can actually be enclosed in this. So it's actually huge content of information. But then if you look at the way the information is organized, it's only 4 alphabets, A, C, T, G, and the combinations of that if you read this. Okay? How do you make sense of it? This information, despite being so large, and it's almost like strings, infinite strings. Okay? Now, for a biological machinery to make sense of this must be really complex. We hear about genes, right? So in this information now, where does the gene begin and where does it end? That's the most fundamental question, right? We say that there are some n number of genes hidden within these 23 pairs of chromosomes. So can one figure out exactly where a gene begins and where does it end? And from that point onwards, where does the next one start? So for the biological machines to make sense of this kind of information, they really need to read this properly. So which means they need to know, let's say if this is a language which we do not understand and each language has a grammar, then the biological machines have to decode the grammar of the genetic code. Okay? How do they do that? That's the most important question that we are trying to understand here. So what I have done is here I have actually extracted the sequence of one gene, okay, and that you will probably not be able to read again. Alphabet starting from here all the way up to here represent one gene. There are few thousand alphabets, okay, and this is one of the smallest genes I could lay my hands onto, okay. So, some of the genes actually can be so huge that you will have to fill probably 10,000 slides with this kind of information content. So, some of them are really large, but this is one of the smallest genes, okay. So, now look at, let us try to understand the grammar of this particular gene. So, this is a gene called Hox A1A, okay, and this is the sequence of the gene. But then can we understand the grammar? So, there are multiple reasons, experiments. I will not go into the details of the experiment. I am just going to tell you that the various color codes that are used here actually highlight different information in this particular gene, okay. So, now you can quickly appreciate that based on the arrangement and the organization of these various elements highlighted in different color codes, you can see that, okay, there is something unique with this one alphabet. It is green, okay. These are purple and these are blue. So, this unique alphabet does something. Then there is a triplet here A, T, G, that is different. There are some other colors here, then again another triplet which is red, T, G, A, and there are large residues in red, and then again some purple here, and then there is one green color here. So, this green and that green, there is something again unique here. So, this is trying to understand and decode the grammar, the way in which one can look at the organization of the gene to start looking at where does it begin and where does it end. Okay. Now, the two green alphabets actually indicate where the gene, so called gene, begins and ends, but then genes give rise to proteins. Okay. The proteins are encoded by triplets of information, okay, that is the genetic code. And if you look at that, then the gene actually ends here, starts here and ends here. So, that is the only sequence that one has to look at. So, the protein coding information from this gene is actually much shorter than the gene itself. Then why should the gene be so big? 3 times bigger than what is the protein coding information? That is another question. So, these are the various questions that we are going to address in our today's discussion, and try to understand the language of genes and the grammar in which one can actually decode this huge information content. So, knowing all this, okay, does it give us the complete picture? Is that really true? Or there is something beyond and above this that will actually enable us to understand this complex language of genes and genomes. Genome is a collection of genes, okay. The picture that I showed you here, this is the genome, this is the human genome, okay. It is a huge collection, it is like an encyclopedia, okay, which has multiple volumes, which are the chromosomes, and each chromosome has multiple genes. And we just try to look at one gene with all these variations, various kind of attributes or characteristics that define the function of a gene. But is that really true? Is it sufficient, whatever is color coded here, is that sufficient to give everything that we need to know about this gene or not? Okay? That is again another complicated question that we are going to address today. So, let us look at now the genomic context. I showed you a picture of human genome, how huge it is. I am going to now show you a picture of multiple genomes, okay, in a kind of a graphical format. What you see on the on the x axis is the size of the genome, okay. It is a log scale and it goes from 1 megabase to 100,000 megabases, okay. 1 megabase is 1 million bases, okay. So, each alphabet here is one base. The bases together are the building blocks of DNA, and that is what makes the spiral of the DNA. So, there are so many alphabets in each of these genomes, and what you see here on the y axis are different species. The bacteria are here, okay. And if you see that all of these bacteria or the very early forms of life have very short genomes, okay, in order of few megabases. But as you go across this, you will see that you have the algae, you have the fungi, you have the plants, insects, fishes, birds and reptiles, etc., and the mammals. And you will see that there is a general trend that the size of the genome goes on increasing. Okay? So, let us say the evolution happened like this, that from early forms of life, more and more complex forms of life evolved. Okay? So, we believe that it may be something to do with the size of the genome and the number of genes, and that is why if you have more number of genes, you get a more evolved or a more complex organism. To a large extent, this seems to be true, but then I am going to give you some examples and some kind of exceptions to these rules that's really going to open your eyes. So, mammals, we are here, we are humans, which belong to the mammalian species, and their genomes are between this size, about 3 to 5 or rather 1 to 5 gigabase, okay, billions of base pairs. Our genome actually has 3.3 billion alphabets, the bases that I told you about. So, that is why we are plotted here. But now, just look at some of the plants. Familiar names, onion, okay. I am sure everybody likes it, but onion genome is bigger than our genome. Okay. Are you surprised? Forget that. Look at paramecium, bigger than human genome. It is a log scale, mind you. Okay. Okay. So, 10 times bigger genome. And if I were to tell you amoeba, you have heard of amoeba, right? It's ugly, ghastly creature, but it's a single celled eukaryote. Amoeba genome is here, 100 times larger than human genome. Okay. So, is it the size of the genome that makes us what we are? I mean, as humans, we think that we are best in everything, and we make a very anthropomorphic statement that we are at the apex of evolution. And therefore, in fact, many people believe that we should have the biggest genome, we should have the largest number of genes and what not, but that is actually not true. Okay? A big blow to you, human ego. Some of the toads, okay, the frogs also have bigger genome than humans. Okay? So, many of these lesser mortals seem to have much bigger genomes than what we have. So, is it then, does it really matter how many genes do we have? Okay, that is the central question. Is it the number of genes that defines how complex as a being we are, or is it something else that defines the complexity of an organism? I mean, undoubtedly, whether we call it as our own view or not, but in terms of behavior, in terms of various other features, human beings are more complicated and complex compared to other species. But we do not have as many more genes as we would like to have. Then the answer to this question is almost meaningless because of the following reasons. So, I am just going to enumerate a few reasons and then spell out by giving examples. So, each gene can give rise to several proteins, because proteins are the ultimate workhorses, they do the work in our cells. So, even if you have, let us say, one gene, it can give rise to 10 different types of proteins by some shuffling of genes. So, therefore, you can increase the number of proteins can do the work. Each protein can then be modified into multiple ways, and these modifications are chemical modifications. Each modification changes its property. These modified proteins can further take part into forming different complexes. So, it is like forming different groups. So, today I am with these two people, tomorrow I could be with these three people, like that. So, different complexes based on different modifications. So, all of this actually completely changes the coding capacity in terms of functional capacity of the genome, and that is why it does not matter how many genes do we have. They are all because of all these modifications and complex formations can actually give rise to multiple functional attributes. So, one such complex is called chromatin, and that is again something very important for today's discussion. And this is a complex of DNA with proteins. Okay? And again, nature is very clever here. DNA, by definition, deoxyribonucleic acid is an acid, right? If you know simple chemistry, there are acids and there are bases. This is an acidic molecule. That means it is negatively charged. So, nature evolved a scheme where DNA gets complexed with proteins which are basic in nature. So, these are histones and they are basic in nature, which means they are positively charged, acids and bases, right. So, just like a magnet, opposite poles attract. So, here acids and bases, they make a very nice complex. If proteins were negatively charged, they will be repelled. So, they cannot come together. So, for them to make a complex, to make a tight bond, you need to have basic proteins. So, nature somehow selected these basic proteins to make contact with DNA, so that they sit together very tightly to form a complex called chromatin. Okay. And the chrome actually means color. So, initially in earlier century, when people were trying to understand structures inside the cell, they used various dyes, and these dyes stained some substance in the nucleus. That is why it was called chromatin, something to do with specific color formation. It is a highly dynamic material which carries a substantial amount of epigenetic information, because it has something in addition to the genes. It has something in addition to the DNA itself, and that is the protein. And these proteins then carry some marks, which are the epigenetic marks. Okay. So, the most important underlying theme here is that all cells in the organism, in every body, carry the same genetic material for a single organism. However, each cell type expresses different genes. And I am going to give you an example, rather more elaborate explanation of how this happens, because that is where epigenetics comes into picture. You are extracting different meanings out of the same material. Okay. The code is same, okay. It's the same Bible that we are using, but the meaning every time you read it becomes different. And that is exactly how different cells actually extract different information content out of the same genetic material. And that is the crux of what we are, because we are different from head to toe. It is not the same structure. Okay. So, this is how chromatin is schematically represented. This is the DNA fiber, which is actually helical, shown here like a simple fiber, and these are the balls of proteins, which are the basic proteins called histones. So, the DNA wraps around each of these almost 2 times, and that is how a very long thread of DNA, okay, and I will tell you how long it is. If you take all the 23 pairs of chromosomes in our cells, arrange them next to each other, let us say this is one long chromosome, this is another short chromosome, keep on adding them, it will be as long as from this fingertip to this fingertip. Okay. So, about 2 meters in length. Each nucleus in our body carries that much of DNA. Okay. And if I tell you each nucleus how small it is, I mean on a pinhead you can accommodate more than 50 of those. Okay. You can imagine how complex this problem is. You have to accommodate a fiber which is 2 meter long inside a structure which is so small. So, how does nature accomplish that? Again, so it is like tying a thread. If you want to take a long thread, wool or whatever it is, and make it shorter, obviously the way to do it is you actually tie it up like this, and that is exactly what nature has evolved. It uses these balls of protein which are basic to tie the DNA around it. Okay. So, you can either make it very compact, pull it together, or less compact. Okay. So, that is the breathing process of DNA. It can either become extremely compacted or it can become open. This is also important, and that is constantly happening inside your body, okay, even as I speak. So, I will again give you examples of how that happens, and I am going to show you a video of how this information actually generates a code which becomes a packing code for the DNA, and that is in the next movie. So, what you see in this movie is actually initially an animation. This is the duplex of DNA, 2 strands in a spiral. Okay. So, this DNA molecule then attracts the basic proteins, which will now, one of them will come and bind here, another will come and make the complete particle like this, and the DNA gets wrapped around it. Okay. So, the DNA is now getting wrapped around, and you will see as you zoom out. So, this is the fundamental particle of chromatin, which is called the nucleosome particle, and then many of them come together to form a spiral coiled coil. Okay? See how beautifully nature packs things inside the body, and you can't see them by naked eyes. These are really very, very tiny structures. You need a very high resolution electron microscope to see this. So, this is called a 30 nanometer fiber because the diameter is 30 nanometers. That undergoes further coiling and it keeps on getting organized. So, it's a series of coiled coils like these, which you zoom out further, and then actually what you will see is a real life situation under a microscope, under a compound microscope, in which visible light will actually show you the chromosomes inside a dividing cell. Okay? Now, what you see is actually live inside a cell. It is no longer an animation, and what you see is a dividing cell. The chromosomes are getting organized across a plate, okay, and this cell is going to divide. The chromosomes have duplicated, they organize themselves in a plate, and then they move towards daughter poles. They are separated, okay, during the cell division. They went on this side and that side, and now the cell divides and you have two cells. So, this is what happens every time a cell in our body divides, and you get two cells out of one. So, the DNA content doubles up, it separates, and the cell divides, you have two cells. So, every time the information content is regenerated. Okay? It is like making a copy, photocopy. You have one, then you make another one, it is exact same copy, you make another one, keep on making copies. That is how the cell number increases. Okay? All of us begin our life as a single cell, and now we have trillions of cells in our body. So, you can imagine how many times this process must have happened in our body to give rise to the entire structure. And during that process, every time it is not just the division, but also differential extraction of information of gene content. So, what you see here now is the various modifications that can happen on the basic proteins. So, these are the basic proteins, and they have all these amino acids which get highlighted. So, what different colors you will see here are the chemical modifications, and these are small chemical groups like a methyl group, a phosphate group, acetyl group. They get added onto each of these residues, and that is how you change their properties, their chemical properties. Okay? So, there is a lot of chemistry happening inside our cells all the time. They get modified, and these modifications basically define the functional state. So, now it is time for me to define what is epigenetics. Okay? Epigenetics is a term that is coined by a very, I would say, visionary geneticist, British geneticist called Conrad Waddington, whose picture is here. He coined this term in 1942, even before the structure of DNA was deduced, even before we understood anything about the kind of organization that I showed you, but just based on looking at some of the patterns of inheritance, he actually came up with this. So, this is a branch of biology which studies the causal interactions between genes and their products, okay, which bring the phenotype into being. Phenotype is what we see, genotype is the genes, okay, which you do not see. Okay. So, genotype gives rise to the phenotype. Phenotype of plants, birds, other animals and us is very different, okay, but the genes could be similar. So, the way he defined epigenetics is these are the heritable changes, which means they can be passed on from one generation to the next generation. These changes in gene expression that are not attributable to the sequence variation. The alphabets that I showed you do not change, because if they change, that is a mutation, okay, and then it cannot be reversed. If we have a mutation in any of the genes, we come down with some disease like sickle cell anemia. One alphabet in one gene changes, and that completely destroys the hemoglobin structure. Hemoglobin is the protein that carries oxygen in our red blood cells. Okay? So, it gets contorted, and the RBC, which is nice round structure like a frisbee, it gets sickle shaped, okay? And then it cannot carry oxygen the way it is supposed to carry. The person dies early in life and does not have a good quality of life, just because of one alphabet change out of the 3.3 billion alphabets. Okay? So, that is a mutation, because the structure, the sequence has changed, however small it is, can lead to such catastrophic changes. Not always, but sometimes. But in epigenetics, the sequence does not change. What changes are these marks, the chemical highlights that you can put onto every protein and its amino acids that completely change the properties in which DNA can be assembled and its meaning can be extrapolated. Another way of showing this, so I just did this before coming here. If you are reading this, let's say you are to appear for an exam tomorrow, okay? So I am reading a book or whatever textbook or some notes from a class, and if I have to remember certain things, what do? I highlight those, right? So then what I'll do is, out of this couple of hundred words, I want to remember only the most important, the key words, okay? Or some people write very important on the side. I have highlighted this, okay? So these are the most important words out of this whole bunch of hundreds of words. I want to remember only 20 words. So I have highlighted them. Has this changed the meaning? They are exactly the same alphabets, exactly the same words. So what has it done? It has just put the same information, identical information, in a different perspective, okay? So by highlighting this, you are just making this more important than the others, okay? Which doesn't mean it is gone forever, it's still there, but you have just highlighted the importance, okay? This is for me. Somebody else reads this, might highlight something else. For that person, maybe this is more important than that, okay? Or even if I were to read this after 1 year, I may highlight something totally different. You can underscore, you can highlight in different colors. So these are the modifications that I am talking about. The genetic information gets modified exactly this, and that is the epigenetic modification. You have not changed the information, you have just changed the way in which you look at it, okay? So these, if you remember this analogy, you will understand epigenetics very well. It's only the highlights, okay? Just the way you change the color of your hair, that's exactly what it is. That is epigenetics. You are just changing the highlights to bring out a different meaning, bring out a different perspective out of the same information. So Waddington actually showed it in a very beautiful manner, pictorially. So what he said was, this is the original picture from the paper that he published, and this is a modern interpretation of that. So imagine, I am sure many of you have played this game where you roll a ball, okay, down some channels, and then you need to move around to direct the ball to go into a certain specific hole, okay? And if you manage to get in a particular one, you get good score, and then you have a prize at the end of it. In his perspective, in this kind of a landscape where there are valleys and there are hills and all kinds of things, let's say this ball is rolling down this, depending upon whether it goes through this channel or this channel or this channel, okay, it can take any fate, any of these channels. It might, let's say this were to be a gene, okay, eventually that gene expression pattern can turn this into either a muscle or liver or leg or eye. These are different tissues in our body. What I will show you here in form of a movie, if you can help me with this again. In this movie, what you will see is, again, this is a landscape, but it's a 3D landscape, and now the movie will show you that this one cell, which is, let's say, no, can you go back? Yeah, just press this, yes. What you will see here is now the cell becomes multiple, divides, the stem cells, and then they slowly start rolling down the hill, okay. That means they are changing their properties, and as they go down in different channels, they slowly change their properties through the epigenetic highlighting, the modifications, and at the end of it, when they are completely differentiated, they change their phenotype, they become completely different, okay. Blood cells, RBCs, okay, versus neurons. See how different they look. Different types of cells in our body look completely differently, but when they all began, if you see the top of the hill, they were identical, okay. Just as I said, we all begin our life as a single cell, a fertilized embryo, okay, that divides until certain divisions, every division is identical, and that is why you get twins. because after first division, for some reason, if they separate and again grow, then you have identical twins, because they actually started from the identical information. Okay? This can go up to the fourth division, but beyond that the cells somehow become different. So, they keep on dividing, okay, and after a mass of cells is formed, slowly they start moving around, they form multiple layers and then they elongate and then they protrude to make limbs and all kinds of things. So, the whole ball of cells slowly changes into various structures. Then you have skin, you have bone, muscle, liver, various other organs that give rise to a complete body. Okay? But if you look at it, all of it began from a single cell. All of these various types of cells have the same genome, identical genome, okay, barring differentiated red blood cells, because they do not even have nucleus, they have ejected the nucleus, but that is a different story, that happens somewhere here. But until the time they had the nucleus, the genome was same as this, was same as that, versus this, all of them have identical genome. They just look totally different because of all the modifications. Okay? So, they got highlighted in different ways, and therefore, they produce different proteins. Neurons will make proteins that are important for transmission of signals, because we conduct impulses, right. If I get pinched here, I will move this aside, because the impulse gets transmitted all the way to my brain through the central nervous system. Whereas, this cell has another function. This is a ciliated cell. Okay? All the time it is in my respiratory tract trying to throw out the dust particle that I am inhaling, because these cells keep on beating the cilia, small hair, which will throw out all the particles and they go upwards, and that is how we sneeze or we have the mucus, etc. So, all cells in our body have different functions, different phenotypes that arise from exactly the same genetic material. So, that is why epigenetics is at the center of studying of all these various things. Stem cells, which are the earliest cells in our body, okay, they can give rise to stem cells again, they keep on dividing, they are all alike, but at some point some switch happens and then they start moving in a different way to make different types of cells. Fibroblasts in our skin, which are elongated. Neurons can be really long, they are the longest cells in our body, some of them are as long as 1 meter, okay, because they go from one end of the spinal cord all the way to the limbs. They are single projections of cells. Okay. So, they have to become like that, whereas blood cells are tiny, so tiny like a circle. So, all of them look alike, look differently despite having exactly identical genomes. These cells then differentiate into various types of cells which look very different than each other, and they can then also become a disease state, and that is why you get formation of cancer, other diseases, and then they eventually also age, okay, the senescence of cells, which is why all of us eventually age. Epigenetics is at the center of all of this. Okay. So, now we are going to understand how epigenetics can control the differentiation, the diseases, and the aging process, because that is what is importance of understanding the mechanisms that contribute to the regulation of the content, information content from genome to the epigenome. So, that is why epigenetics has been the most, I would say, important revolution in modern biology. You see multiple different magazines have covered this, and with very attractive titles. So, on Time magazine there was a title, "Why Your DNA Isn't Your Destiny." Okay. We believe that DNA is our destiny. We are born with genes and we stay with that, and that is what determines our destiny, but no longer. Now, we know that DNA is not our destiny. National Geographic published a study on twins, which actually showed that with lot of data, that if twins are raised under completely different environment, they look different, they behave different, they do all kinds of different things. So, it is the environment that seems to play a role there. Okay? That is one of the most strongest evidence for the theory of epigenetics, that environment can also play a big role in terms of describing the activity of genes. So, it is a very, I would say, poignant title, alike but not alike. Okay? So, their genes predispose them to be alike, but actually the environment does not make them look alike. So, what is the environment's contribution then? Okay? This is another book, very nice book. If you get a hold of it, you should read this, The Epigenetics Revolution. It's a revolutionary new concept in modern medicine and science, and that's why we are going to talk about what this is. I'll give you another very striking example of epigenetic regulation, and for this I will talk about bees. All of you must have seen honey bees. You may like honey, but I'll tell you what about something very unique about honey bees. Honey bees have different castes based on the work that they do. You have worker bees, you have nurse bees, you have forager bees. They have very well defined tasks, okay? And then there is a queen, again a very defined task. The task of the queen is to lay eggs, that's it, okay? There is only one queen per hive. All she does is basically just lay eggs, hundreds of eggs per day. Workers, they are the hard working people, they are the hard working community, thousands of them are there per hive. All they have to do is keep on building the hive, go out, fetch the nectar, make the honey, okay? And the nurse bees have to feed royal jelly to the young ones, nurture them, okay? So, these are multiple in numbers per hive, but there is only one queen, okay? Though they are different looking bees and doing so many different functions, well defined functions, their genome is exactly identical. And people have done these studies. In the last 5 years, a lot of epigenetics research has been done on various insects, including ants, bees, wasps. They are called social insects, okay? Just like humans, they also have a lot of social characteristics. They have colonies, they have hierarchy in their social structure. So, it is very interesting to study, because in humans you can't do this kind of studies. We can't do experiments on humans, but on ants you can do experiments, right? If ants are walking around, you can lift one and put them behind and see what happens. Or in this case, what they did was they actually removed the queen bee from the hive. So, the hive cannot exist without the queen, okay? There is no question. So, then what happens is one of the worker bees actually turns into a queen, okay? Just one, not two, not three, not four, just one out of the 1,000, okay? It is a very, I mean, mind boggling. How come only one worker bee decides or can be changed into the title and become a queen, and not just change the title, that bee also becomes 40% bigger, okay? So that so many eggs can be held in the abdomen, because if the same size you can't lay so many eggs. So, there is a change in the phenotype also without change in the genotype. The genes do not change at all. Suddenly there is a transformation and only one worker bee becomes the queen. There is research that has actually told us that some epigenetic modifications happen and that change a worker into the queen bee. And same thing, you can actually interchange between the castes in the bee just by changing their epigenetic modifications. And you know how, what changes those? It is really amazing. I mean, they discovered epigenetics millions of years ago. We are only finding it now. It is actually through the royal jelly that they feed, okay? So, the worker that has got during early childhood, during their development, more of the royal jelly, okay? The royal jelly contains a lot of nutritious substances, but also some epigenetic modulators, small chemicals, okay, that are there in the jelly, which go into their body and make their epigenetic makeup. So, out of the thousands of workers, the one that has got in the childhood more of this is the one that is destined to become the queen if there is absence of the queen, okay? The choice is already made based on the food, okay? So, this is really intriguing. How does this work in nature, that all these epigenetic changes actually predetermine what these bees are going to do, okay? And by changing that, you can actually change their fate, okay? You can make forager into a nurse, nurse into forager, or workers into queen and queen into worker. Just like that, okay? Just change some of these highlights and they completely switch their caste. So, it is amazing. I mean, that is one of the most striking examples of epigenetic regulation, where you can change phenotype with epigenetics without changing the genes at all. So, that is how despite having limited number of genes, and now I will give you the numbers, we have only about 20,000 genes in every cell of our body, okay, despite having such large genome, 3.3 gigabases. And that number, mind you, is not very different than the number of genes that the fruit fly has. Okay. The mice and rats, which are very extensively used for animal experimentation, have exactly the same number of genes as what we have. Okay. So, it is not the number of genes that defines what we are, it is the way in which they are regulated and the way in which they are used that defines what we are. Okay. So, the finite, it is a really small coding capacity. Now, no longer anybody talks about the genome size, it is futile to talk about the size of the genome. Okay. You cannot boast it by saying that I have such a large genome, because it doesn't matter, nobody cares what is the size of the genome. What actually is important is how the finite coding capacity of the genome can be extrapolated into bigger and bigger coding capacity. It is like taking the projector away or the screen away, so that you get bigger and bigger projection size for the same projector. So, the finite coding capacity of the genome can be extrapolated into much bigger coding capacity through all these effects of environment and the other cues that the cells acquire as they are developing to make into a much larger coding capacity, which we now call as the epigenome. Okay. And that is precisely why, despite having very similar genomes, all of us sitting here in the auditorium look so different than each other. I am sure nobody will claim that I look like the other person. We all admit that we are very different than the other person, not just in our looks, but also in terms of that is phenotype, but also in many other ways, behavior for example. Okay. Our likings to many things, from food to perfume, whatever it is all part of the way in which genomic information gets extrapolated into our phenotype. Okay. And that is what is important, how this happens, and this is where the environment plays the biggest role. Okay. How finite coding capacity becomes almost infinite is through the constant interactions with the environment. So, in the next, what I am going to talk about is these examples of how the environment influences genes, and I am going to take examples of diseases which are called NCDs, non-communicable diseases. Okay. And why non-communicable disease? Because communicable diseases are easy to understand. There is a virus, you get infected with the virus, you come down with flu or some other disease. There is a bacterium, you get infected with the bacterium, you get an infection. Okay. So, it is a simple cause and effect relationship when it comes to infectious disorders, right. When it comes to non-communicable disorders, there is no simple cause and effect association, right. You can't say that I was in a polluted city, living for 30 years in a polluted city, and that is why I have lung cancer. Can you say that? You can't say it very confidently, right. Or because I eat lot of jalebis, I got diabetes. No, you can't confidently say this. Maybe that contributed to it, but that is not the definitive answer or cause. So, this is a very complex disease to understand, because it is non-communicable. These are more complicated or complex disorders. And I have again taken examples to say, actually as fathers, many of us have done lot of sins. Okay. And this is actually from a scientific journal, this is from Nature. Okay. There was an article which, so I have just taken the picture straight from this is the reference in 2014, sins of the father. What can the father do to mess up with the next generation? Okay. So, I am going to give you 3 examples. So, beware all fathers. The first example, okay, this first example is about influence of diet. Okay. And here in this case, again a paper from Nature, very respected journal, and what you will see is this is the effect of chronic high fat diet. Okay. Very important topic. So what they did was, so rather I'll tell you what is the question. The question is, can diet cause heritable change in physiology via epigenetic changes in gene expression? Okay. So they took a group of rats, experimental animals, fed them with high fat diet, and chronic means for several months. Okay. So if you feed them for 3 months, let us say, that is equivalent to 15 years in human life, because rats live only for 2.5 years on an average. Okay. So 3 months is equivalent to about 15 years of our lifetime. So it's a chronic treatment. So you get them used to high fat diet for few months, and then look at what happens to their progeny. Okay. What they actually found is that, I'm not going to show you any data because it's not important for this. They actually found that fathers who gave rise to progeny after being exposed to the high fat diet, actually their daughters, female offspring, had beta cell dysfunction, which means these beta cells are the ones in pancreas which produce insulin. And I am sure all of you know what is the function of insulin. Insulin is a hormone which is important for absorption of glucose in the blood. Okay. So that is why when you have any test for diabetes or something similar to that, they will first test blood glucose level, okay, which is an indicator of insulin function. Okay. So this that means they were predisposed for type 2 diabetes, okay, without doing anything. They ate normal food, okay, they did not eat high fat diet, but what had happened was their fathers ate high fat diet. So this is quite alarming. I mean, by doing something in one generation, the next generation is suffering, which is something unheard of. Okay. So this is actually from again the story that I highlighted the slide before. This is the conclusion of their paper. Fat fathers affect daughters' health. It's been experimentally shown. The second example, here the perturbation is not of diet, the perturbation is of other environment, and this is again very interesting experiment. It's a very complex title. I don't want you to read this just because I am giving you the real cases. I took it exactly from the paper which was published in Cell in 2010. There are many papers after that, but this was the first one. Okay, that's why I have chosen, just 8 years ago. So what they showed is that if you change the environment in such a way that you provide stimulation to a mind. Okay, now this is a more complex question. Does brain activity and state of mind influence gene expression and eventually our health? Okay. This is again something very important for the modern lifestyle that we all live. Forget about diet. Now it is talking about state of mind. How do you measure that? Okay. So what they did was, experimentally it is slightly difficult to quantify, but there are ways in which one can do that. So what they did was, they put again rats, favorite experimental animal, into, normally they are kept in small cages. Okay. If you go to any lab, they have animal house where if you go, they are kept in nice cages, they are contained, and you have typically about 3 to 4 rats per cage, which if you see is about 1 square foot area. Okay. So it's very tiny area. Normally rats and mice, they are rodents, they like to explore, they want to go here and there. If you let them loose here, all they will do is run around everywhere, keep nibbling, and that is what they do in real life, right, in the wild. But if you confine them in a small cage in the lab, they don't like it. They are completely bored to death actually. They are frustrated and they have nothing to do. Okay. Sometimes they become aggressive just because of that confinement. They start biting at each other, they start fighting with each other. So this confined environment versus an enriched environment, which I will show you some experiments that were done in IISER. Some of the IISER students did this part of the experiments in a different scenario, but I will just show you. This is exactly what the authors also used for this particular paper. So you keep a rat's version of a treadmill, they could do some exercise, they could run in this treadmill. Okay. It works very nicely. You can see the rats are running in the treadmill, and the cage is much bigger. You can see 1 meter long instead of being a small cage. Now it is very big cage. You see from top, there is a treadmill here, there's a tunnel here, there are all kinds of other things where they can explore. Okay? So, you may give them a slightly more area to play with. A toy, multiple toys, you see the rat is now going to go through this tunnel, and every day you keep changing this. Okay? So, it is not a predictable environment for them. So, every day there is something new to do. So, that gives them an enriched environment. They are stimulated rather than being bored. Okay? So, you create these two experimental systems, and then of course, you can add all kinds of enrichment, textured surfaces, different smells, everything that they explore, get to explore in the wild. You can try to mimic that, and then you do this again for 3 months, bored environment versus enriched environment. Okay? 2 groups, don't mix them, one group in the same cage, another group in this enriched cage, and then after getting them used to this kind of environment for 3 months, which is again a long span of their life, the experiment that was done by these people was to inject highly aggressive tumor forming cells through the tail vein. Okay? So, they go through the blood circulation and they lodge into the peritoneal cavity in the abdomen of the rats, and they make tumors. Okay? So, normally this is an experiment, this is called a xenograft, where you graft cells from a tumor into a normal individual, and that makes a tumor. Okay? So, you grow tumors like this in the control, bored animals, enriched environment containing animals, and then look at what happens to the tumor formation. And very strikingly, what they found is that in the animals which were exposed to the enriched environment, tumors regressed. In fact, they did not see any tumors, versus the control rats which were in the frustrating, boring environment formed these tumors. Okay? And then they characterized the entire gene network circuitry that led to this. Sensory stimuli that goes through the environmental enrichment through all the senses, smells, vision, sounds, everything put together, tactile sensation, the touch, etc., all put together somehow enriches certain genes in the brain. The arrow indicates they are going up, they are increased, the stimulation, and that somehow feeds through the network of fibers. Okay. The names are again not very important here, to the rest of the body, affecting its physiology in such a way that another gene which is called leptin is down, okay, and that leads to the remission of tumors. So, just because of this environmental enrichment, now your body somehow can suppress formation of tumors. So, this is again a very profound conclusion, that somehow having an enriched environment around you can reduce your ability to make tumors, which means it makes you resistant to tumors. That does not mean you will never get cancer, but means that you actually can lower your ability to have cancer. So, let me just talk about the genomics revolution, how this is important and how new revolutionary technology can actually assist in this kind of experimentation. So, one of them is the sequencing. You have heard about the genome sequence, human genome sequence project that was done using the classical sequencing technology. Now, we have the second and the third generation sequencers. So, these are the two ones that we have used. This is a phased out technology, outdated several years ago. We still have the ghost of this machine, it does not run anymore, but it is quite big. It is about 6 feet tall and 6 feet wide. The fluidics, this is a supercomputer here to analyze the data, and other things are here, and this is the display. One can use this and sequence human genome in few months. Okay? In one lab, one person can do that. That is the power of these machines. The classical technology which was used to sequence human genome in the US and other countries, which cost about $3 billion, the so called human genome project, which was akin to the project that took man to the moon, really large project, took more than 10 years and thousands of scientists. Okay? So, you can imagine from that to be able to now do the entire human genome in a few months time frame, and that too by just 1 or 2 people, was achieved by this next generation sequencing. So, this is called the next generation sequencing. But technology doesn't stop here. Now everything is becoming smaller and smaller. Okay? So the next generation beyond this is actually becoming even more small. Okay? And I'll tell you, I'll show you the picture of the next generation sequencer, which is now going to be so common that eventually even pathology labs will have these sequencers. Okay? That's the prediction, because they look like this. Yeah, this is my hand, and that is actually just like a small stapler. You know, this sequencer, you can plug it with a USB 3 cable to your laptop and you can start sequencing. You know, it's the way in which technology is changing is really alarmingly rapid, and this finishes the work in like few minutes. Okay, in 20 minutes the entire run is over, as opposed to weeks of time. Okay, so it's really rapid now, and if you see the costing also has dropped dramatically. This slide is particularly important because many of you are aware of Moore's law. Okay, this is the Moore's law. The sequencing field has beaten even Moore's law. Okay, the cost of sequencing has dropped dramatically after the invention of the next generation sequencing technologies, and you can see that this is where it, up till this point, it is following Moore's law, and now beyond this, in the last decade, it has fallen further down, and that's why sequencing your own genome is going to become a commonplace. I have data only up to 2015. If you extrapolate it now to 2020, by next year onwards, yes.
Yeah, so can you please characterize on this graph what you mean by first, second, and third generation? What you were showing earlier?
This is the first generation. Okay, the classical sequencing.
Steep is second one.
Yeah, and now there is a third generation coming. So you see, these are $1 million. Okay, upwards of $100 million for one genome. Now the cost is coming down to $1,000, and eventually it will be actually just few thousand rupees. Forget dollars. Okay, so 5 years from now, I can imagine all of you will carry your genomes in a stick, go to the doctor, he'll plug it in the computer and look at your genetic kundali and say, okay, this medicine doesn't work for you, you take that medicine. It's going to happen, believe me. We are progressing at that rate. Okay, this is really going down, and because of the sequences, the kind of sequencers I told you, you don't need a fancy lab to run this sequencer, right? You can run it in your backyard or in a small pathology lab. It's that simple, because the chemistry, the way in which data is analyzed, the software, the algorithms is changing every day. Okay, so one has to also keep up with this kind of technology. So this is good for medicine, because then we are going to go more and more towards personalized medicine, because this is going to help us in terms of having genomic information and then decide whether one kind of therapy is good for this person versus the other person. So I'll just show you some technical things, because specifically for the audience here, I thought this will be important. The amount of maths, statistics that has gone in this particular technology and the algorithms is quite amazing actually. And this is where biology has been helped by other fields to develop these technologies other than the chemistry. I am not going to go to the chemistry of sequencing, but this is the analysis. So you can see that every sequencer throws out files like these. Okay? So not just the sequence, but the large amount of other data, which is basically something to do with that particular sequence file. Okay? And you get millions of these out of every run. In fact, these days billions of these. So it's a huge amount of data. We are talking about big data now. Okay? So if you collate all the sequencing data that is accumulated every day in the world, it's astronomical. So again, you have to devise algorithms for analysis of this kind of big data. So people are actively working on this, how to collate all of this together and make sense of it. Imagine you have data sets of billion file reads like this thrown at you every day. Okay? How do you make sense of it? So to give an analogy, decoding a sequence from this kind of sequences, genome sequence of 3.3 billion, the human genome, from short reads like this, which are 100 base, is like taking a book, putting it into a shredder, okay, getting all the shreds, and then reassembling the book back. It is as complex as that. Exactly that's what we do. The algorithms that we use for sequence analysis enable us to take these shreds, which are the smaller sequences, only this much, and assemble the entire genome. Imagine you have only 100 bases. We don't know where the next one comes from, right? It's not next to it in the genome. This is how the sequencer gave, but these are random fragments. Okay, this is called shotgun sequencing. So you are taking random pieces of the genome and giving them as strings. You have to put them together as if it's a continuous string in the genome. So that is like this. You have to reverse this process. How do you do that? So very complex algorithms have been evolved to do this. If anybody is interested, I can give you the algorithms that are used, but they are really state of the art and complex algorithms that have been used. So in the last 5 minutes, what I'm going to do is talk about the human context, because that is where all of us, I think, would really like to use this information of epigenomics. So we all know about evolution, how we evolved from apes, and there are multiple theories, but now people believe that really this is how we evolved. The question is, has the evolution stopped? And obviously not. It's an evolving process, right? How can it stop? Because that will be the end of life. So life has to continuously evolve. So where do we go from here? So now we have obviously evolved further, and we are here now, right? So social media has completely changed the way in which we interact, and we also be. Our being itself has changed dramatically because of social media, and not just social media, because of various changes in our lifestyles. Also, we have changed in terms of our work habits, in terms of our dietary habits, in terms of everything. Okay? So we have continued to evolve. Now the thing is it really going to help us or not? How do we make sure that we actually can take care of our health as we continue to evolve in the modern life? In fact, Aldous Huxley said in the 50s, medical science has made tremendous progress, so much that there is hardly any healthy human left. He should have said it now actually, but he predicted it long back. So despite all this progress that we think in medicine, actually the number of unhealthy individuals has increased, and that is the bane of modern life, that despite having so many medicines and knowing so much about cells and DNA and protein and whatnot, we actually have more burden on health. So what has caused this? So one of the way in which one can look at this is just look at the disease propensity. Okay? So this is called World Mapper, and you actually can go to this site and ask your favorite gene or whatever disease, and it will give you a map of the world in a very distorted way, and the distortion is proportional to the burden of the disease. Okay? Diabetes. I am sure all of you remember the world map, how it looks, and look at the distortion now. Look at India. Okay? We are bloated into a big balloon. This LBW is nothing to do with cricket. We would still be like this, but this actually stands for low birth weight babies. Okay? When babies are born in India, they are typically 20 to 30% low birth weight compared to the Western counterpart. Look at the Americas. Okay? This is proportional to LBW. Their babies are born with the ideal weight. Indian babies, very low birth weight. That's why the country's size have bloated up. Same is true with diabetes. Of course, developed world also has more diabetes for other reasons, but in case of birth weight, they are doing pretty good. Death from non-communicable disorders, again, look at India. So take any health parameter and put it there, you can do it yourself, you will get the same picture. India is doing very well, okay, unfortunately. So why has this happened in our country specifically? Okay, have we asked for it? So to address this partially, what one of my collaborators, Anand Hardikar, who was in Pune, but now he has moved to Australia. What he did was he tried to understand, can diet be one such factor? And I'll take you through this and then take to the experiment. Are the improper diets the risk factors for diseases. So, if you look at the food pyramid, this is the food pyramid, diet and nutrition actually feeds into this epigenetic circuitry through its effect on a process called transcription, okay, which converts the information content of DNA into RNA, and then RNA is translated into proteins. Proteins are the workhorses that do the metabolism, okay, the enzymes, etc. And then finally, you have a functional cell, okay, that is the normal function of a cell. But what is known now is dietary compounds can affect gene expression and also the epigenetic part of it, okay. And that is why it becomes important to understand the science of nutrigenomics, how diet influences gene expression, okay. So, diet will ultimately affect the epigenome, and because of that it will ultimately have an effect on the phenotype, the being, and then finally that will affect, that will give rise to either health phenotype or a diseased phenotype, okay. So, it is intricately linked with the connection of diet with the epigenome. So, to address this, what Hardikar did was he proposed that by changing diet over generations, one can affect the propensity for non-communicable disorders. So, he proposed this and did a very heroic experiment. This whole work took 13 years. Okay. So, to basically prove this hypothesis. So, and again coming back to the world mapper images that you saw, why India is the hotbed for all these non-communicable disorders? Probably it has something to do with what our forefathers did. Okay. So, to recapitulate that, so if you go to historical reasons, then you will see that typically, I mean the masses in India have been malnourished, okay, for whatever socio-economic political reasons from 18th, 19th century onwards. So, for 2-3 centuries the entire population, I am talking about masses, the 80% population has been undernourished. Okay. And now look at what has happened in the last 30 years. Because of globalization, whatever, liberalization, whatever you call it, everywhere you have increased access to high calorie diet. Okay. I have been to the remotest part of the country and I have not found any shop in the corner of the village that does not sell carbonated beverages or chips. They will not have lime juice, but they will have all these drinks, okay, the soft drinks. And it has happened dramatically, especially in the last 2 decades. The entire country has been swamped with all this kind of food. So, in the last 2 generations, 30 years is about 2 generations, there is sudden access to this high calorie diet for the bulk of our population. In fact, that is what is the food available to the remote villages. In cities at least there are other joints, but in remote villages if you go, every shop owner has, and I have pictures of this actually. Yeah, it's alarming that the access has increased so much, and that way our country has become unified. You go anywhere, you will see all the same brands anywhere you go in the country, small village, remote vasti to big cities and small cities, everywhere. So, to recapitulate this, this experiment actually involved multi-generational undernutrition, and that was done for 50 generations, 50. It took 12 years just to establish the model, because every few months the rats will reproduce. So, you do this for 50 generations, and what was done was, instead of giving regular food, they were given only half the quantity. So, it is like if you eat, let us say 4 chapatis or rotis per day, you will be given only 2, okay. No difference in quality, only quantity was reduced to half. So, the rats got exactly half the calories per day. It is like rationing the food, and then after doing this for 50 generations, one after the other, serial generations, okay. So, the grandfathers, their grandfathers and their grandfathers, all of them got less calories, and followed by this 50 generations, 2 generations of regular diet, okay. They got not exactly more diet, exactly the same diet, no high calorie diet, regular diet. So, that is called dietary recuperation, okay. in the 13th year. That's when the experiment is the model is complete. Then you do the experiment. Okay, you generate the model, and then you do the experiment. So you get to do the experiment actually after 13th year. Okay, so you have to have the patience to do this across half of your career. Okay, so that's the kind of heroic effort Hardiker did, and then I jumped into the picture to do the epigenetic analysis, and this paper was published few years ago. Raised quite a flutter because of the profound implications. So I will just show you the model. This is the control rat, undernourished rat. Okay, can you see the difference in the phenotype? Okay, just counterintuitively, the undernourished rat actually was more fat. Okay, the belly fat, as you call it, pot belly phenotype, is because of that. See the change. There is more girth here than this one. Correct? Now I am going to show you. So of course, if you see the weight, body weight, overall body weight is low initially, but later on they catch up. Okay. If you see these two graphs, this is the control rat, undernourished rat, they catch up the growth. Okay. But eventually, initially they are slow, but over time, once they get old, they are almost of the same body weight, but the distribution changes. What is even more alarming? Yes.
So is this the observation on the in the 13th year of the in the 50th generation, or?
Yeah, undernourished in 50 generations. Okay, but you start seeing differences after the first few generations. Okay.
So the age in age is again for 50th generation.
For the 50th generation, yes. What is alarming is this. Look at the difference. This is R2, 2 generations of regular diet. Okay, they become even more obese. Okay, now to prove this further, we did MRI for the rats. You can do that. There are smaller MRI machines. Look at this. This is the MRI of the rat. Okay, and what you see in white color, the white patches are the fat in the rat. Okay, control, very less fat where it's supposed to be, little bit. That's it. Undernourished. Look at this. Has already got the belly fat. But then look at the recuperation rat. It has fat all over, including the head. Everywhere, the whole body is full of fat. And they ate regular diet. I mean, it's such a stark effect. These guys actually ate something which is supposed to be right. Okay, they did something very right, but they became obese. 30% increase in the body weight. Look at this. Complete change in the phenotype. And not just that, even the distribution of fat changed. So their only bad luck is that they had these forefathers. Their forefathers did something wrong for no fault of theirs, but the next generation is suffering. And that's exactly what is happening. I mean, whatever increase in the incidence of non-communicable disorders that you see is probably because of what our forefathers must have done. But can you just blame them and say that, okay, I don't need to do anything? No. The good thing is these are not genetic effects. Epigenetics is the silver lining because it is reversible. Unlike genetic defects, which are non-reversible, okay? People are still trying to do gene therapy, it has not succeeded. It's very difficult to change the genes, but you can change epigenetics because all of the highlights can be reversed, you can erase them. Okay? There are biological mechanisms to do that. Over time, with proper exercise and nourishment, one can actually do that. And that's the essence of this entire story. I will not show you all of the data because it's not necessary, but it actually changes the epigenetic modifications. That's exactly what we showed. In these models, the only difference is the epigenetic modifications. Because their genome doesn't change, they have only eaten some amount of food, less or more, that's it. Genome should not change. What has changed is the epigenetic modifications. So all of this put together, you can now see how beautifully it comes together. The physiology of the organism depends a lot on the environment, and it has all these small effects, okay, on the function of the muscles, function of the brain, rest of the body, and somehow because this is transgenerational, it got carried from one generation to the next generation. Fat fathers affected the daughters, right? Stressed, frustrated individuals affect their progeny. Yes.
Sir, according to this inheritance or transmission of characteristics, it's only the genome, the genes which get transmitted, right?
Yes.
And if the genes are not altered, how does one generation's whatever behavior get to the next one?
Yeah, so that is my parting shot. What I'm going to tell you now is, if you remember the abstract, the last sentence, you pass on more than the genes to your next generation. And it's a little scary also, because all your lifetime's experiences, okay, somehow get added to your genome. And what is shown here in this picture, actually, this is the mouse sperm, okay. It looks slightly different. It has a hook in the front. So, this sperm actually contains all the smaller molecules in addition to the DNA. These are called microRNAs, okay, and they actually transmit even some of your lifetime experiences, like for example, the fear, the anxiety, depression, all of that gets passed to the next generation, okay, and that has been experimentally proven. So, it is not just your genes that you pass to the next generation. Many of these experiences and your likes and dislikes may also get passed to the next generation. So, it is really, I mean, mind boggling. How does it happen? And which indicates that you need to fix this first. You have to be good in one generation, and only then you can create the better next generation. You can't do it other way. So, that is where I summarize. This is the take home. What we have talked about is the environmental factors, including diet, have profound influence on gene expression patterns, which are governed by epigenetic modifications, including various modifications such as DNA modifications as well as histone modifications. What is important is that the period of exposure or the treatment during life of an individual plays a major role in determining the outcome of the diet induced changes. The silver lining, though, is that reversibility of such changes can be exploited as a strategy for therapeutic interventions targeting the epigenetic machinery. Okay? So, which means one can now think about, and in case of cancers, people have already done this. There are multiple epigenetic modulators in clinical trials already. Hopefully, in the next few years, we will have epigenetic therapy for cancers. For other non-communicable disorders, research is still going on. But eventually, you will have epigenetic therapy for many of these diseases to reverse all these ill effects of environmental insults and diet induced changes. This is a beautiful illustration by one of my talented PhD students, Neha. What she actually showed it is a very beautiful summary of what I just said. There is a life within life. Okay? This is an epigenetic circle of life. And again, this is based on some hard evidence, which is shown that this is a baby within the womb of a mother, and you can see this tiny baby inside this baby. Okay? So, this is the third generation, right? So, even this is affected in the womb of this mother, okay? And which means that even the tiny precursors of the ova, the ovum in the ovary, which is not yet fully mature, of this female fetus which is inside the womb of a mother, also gets affected by the effects on this mother. Her stress, anxiety levels, her nutritional status can affect not just the baby in the womb, but also the baby within that baby. Okay? So, the effects are far reaching, and people have actually demonstrated epigenetic changes in this tiny ova inside the ovary of the baby in the womb, and that is why this picture comes from. So, you can actually see that there is life within life, and these changes are actually quite far reaching across multiple generations. So, the ideal time window to make the change is when the baby is in the womb. So, if you can somehow increase the quality of the environment here, that is where you can make most of the difference. That is why the nutrition and the lifestyle of the mother becomes most important for the benefit of the next generation. So, I stop here by acknowledging the people who did this work. This is my team, and collaborators who have done helped us to do many of these studies, and of course the funding support that comes from various agencies. This is the Center of Excellence in Epigenetics, which is through a grant from Department of Biotechnology, and I thank you for your attention.